This comes to a head that I think kind of speaks to my ignorance. So for the longest time, when I pictured cancers being able to, hide in plain sight, I've always thought of it more like they wore a cloak, or like the the inhibiting functions on them. I thought it was more like something they would have on them, but it sounds more like if you can take something out of the blood, that it's more like a like they emit the chemicals that more like a bath, like they kind of create an environment around them, but they can't be seen. So it sounds like it's more like the second than the first, or at least like a combination. And shows like my ignorance of the science of this. But I always thought it more like you had to find them, and then you had to bind onto the site, and then you attack them that way. But if you're able to just filter the blood out from the chemicals that are hiding them, and then they're able to be seen, it kind of seems like it's more the latter. Like they're not like having. If that makes sense, I feel like I'm talking a little bit out of like not
No,
very well on this.
actually, what you're saying is pretty much aligned with current dogma on
Hmm.
how cancer works. Even most serious cancer researchers view cancer's defense as being primarily a membrane mediated defense. So you have molecules on the membrane of a cancer cell that inhibit incoming immune attack. That's kind of dogma. It's not correct though, or it's not sufficient. It's not complete. So we actually know there is literature. There's substantial research, even though it's not yet dogma. There's substantial research that shows that tumors use a combination of inhibitors. They use membrane inhibitors, like right on the surface of the cancer cell, but they shroud themselves with a gradient of soluble inhibitors. So it's most dense in the tumor microenvironment, less dense in blood surrounding the tumor, and less dense still in general circulation. So this is actually analogous. I use a lot of military analogies because I really feel like in cancer, it's a war. It's a war between the tumor and the immune system. So the analogy that's most relevant here, we call it the bullets and bayonets analogy. Any soldier would prefer to use bullets before bayonet because by the time you have to use a bayonet, the enemy's right on top of you. It's a last ditch defense. Likewise, in evolutionary biology, the evolution of these membrane immune inhibitors is the last line of defense. And again, cancer just hijacks methods used by the fetus or the placenta. So the placenta has membrane immune inhibitors as its last line of defense. But before those get engaged, the last thing the fetus wants is to defend itself against immune cells that are right on top of it. It wants to neutralize the mother's immune system at a distance, but only in a boundary region. If the fetus neutralized the mother's immune system totally, systemically, the mother would die of whatever infections are floating around. So evolutionary biology had to come up with an arbitration between two incompatible survival advantages, one being having an active immune system, and the other being carrying young internally, right? Those are two great survival advantages. They both came up a couple 100 million years ago, but they're not compatible with each other because if you're carrying young internally and you have an active immune system, those are incompatible. So you need some sort of way of arbitrating between the two. And what evolution came up with was local immune inhibition in the region right around the placenta with a mix of soluble immune inhibitors to neutralize the immune system at some close distance, not systemically, but at some close distance, with a high concentration gradient of soluble immune inhibitors. And then as a last ditch defense, you have membrane immune inhibitors. And cancers just hijack that. It uses
Mm.
the same basic approach and the same molecules, the same specific molecules. And then I would say there's a further, you spoke of hiding from the immune system. Some of the immune inhibitors that cancer secretes, that the placenta secretes, are blinders. So they actually bind and block specific proteins on the surface of immune cells that are detectors that detect stress proteins. And you can think of that, there's a military analogy for that would be radar jamming. So they're basically jamming the radar of an enemy bomber, right? That's only one class of immune inhibitor. There's also immune inhibitors that shoot down the bomber. So there are molecules that cancer cells secrete and that the placenta secretes, that if an immune cell gets too close, that cell will, they're not generally killed, but could be killed, but they'll definitely be put to sleep. Put into anergy is the term. And that's the second category of immune inhibitor. And then the third category, which is the one we're focused on first, you can think of as a missile and an anti-missile missile. So all immune cells, pretty much, most immune cells, let's say, have one of their main cytotoxic swords is a molecule called TNF alpha. So they'll deliver this molecule focally against an immune cell. It can kill that cell directly, and it also can activate and recruit other immune cells to join the fight. It's a general pro-inflammatory cytokine or signal molecule, okay? What cancer does to defend itself against that incoming attack, in other words, the cells are now irrelevant. The cells have launched this highly potent cytotoxic missile, if you will, against the cancer cell. How does the cancer cell defend itself against an attack that's already underway? Killing or repelling the immune cell at that point wouldn't do the trick. So what it does, and all cancers do this, all solid tumor cancers do this, it cleaves off the extracellular, the portion outside the cell, the receptor, the antenna that receives that death signal from TNF alpha. And that receptor is called a TNFR or TNF receptor. And what cancers do is they cleave it off so that it leaves the cancer cell, but it still maintains binding affinity for incoming TNF. It'll bind it and neutralize it like an antibody. But then the signal, the death signal from that incoming TNF, can no longer be transduced into the cancer cell. And the cancer cell is now protected. It continues to grow and continue to make these TNF receptors and cleave them off and into a very high concentration bubble of protection, a protective shield inside the tumor microenvironment. And that's the target we're going after. These soluble TNF receptors, they're called sTNFRs for soluble TNF receptors. If you take those out, then immune cells can now deliver TNF and directly against a cancer cell and destroy it. And that's what we've proven works in animals. And also there's a group in Germany that does it by pheresis and has documented the highest efficacy ever recorded against more than 25 different cancer types. So that's our first product that we're going to bring to human clinical